Welcome to CFRI's Cystic Fibrosis Community Voices, a video podcast series created by and for the cystic fibrosis community.
Hello, everyone, and welcome. I'm Siri Baith, Executive Director of CFRI, and I'm really honored to welcome you to what promises to be a very informative presentation with Dr. Jim Chmiel, who is a national expert in cystic fibrosis and anti-inflammatory therapies. Before we get started, as always, I want to remind people that the information presented during this podcast should not be used for your own medical advice, diagnosis, or treatment. So if you are pondering making any changes in your medical regimen, please consult with your team. This podcast is possible thanks to the generous support of our sponsors, Corbus Pharmaceuticals, Gilead Sciences, Genentech, Chiesi USA, and Viatris, with additional support provided for today's presentation by Corbus Pharmaceuticals. For those joining us today, you can ask your questions throughout the presentation by using the Q&A box. Dr. Chmiel will address them at the conclusion of his presentation. For those of you who are joining us via Facebook Live, welcome. I do apologize, we are not monitoring those questions. But this podcast it's being recorded and will be released within the next 2 weeks. So now, on to the substance. It is my honor to introduce Dr. Jim Chmiel, who's the Division Chief for Pediatric Pulmonology, Allergy and Sleep Medicine at Indiana University School of Medicine. Dr. Chmiel's research focus is on airway inflammation, especially as it relates to cystic fibrosis and asthma. And he was the principal investigator of Lenabasum, which many of us have been watching with anticipation as it works its way through the pipeline. Dr. Chmiel will review the results of the recent Phase IIb trial and its implications for our CF community. Welcome, Dr. Chmiel, and thank you for sharing your time with us all. I'll let you take it from here.
Great, thanks, Siri, and thank you for the invitation. It's an honor for me to be here today and share the results, and certainly we'll be happy to answer questions at the end. And so we'll go ahead and start the screen share. So I am going to review the results of the Lenabasum Phase IIb clinical trial known as JBT 101 CF-002. As far as disclosures, I received funding from the CF Foundation for consulting, but also research funding from the CF Foundation, the NIH, and industry sponsors for trials that I participate in. That funding doesn't go to me personally, but to my institution, and Corbus as well supports the research effort of this clinical trial with funds to my institution, but nothing to me personally. So I want to just review inflammation a little bit and to kind of get us set up here. CF is a condition which there is a chronic inflammatory process. Once the inflammatory response gets activated, it continues to rise, and then it is excessive relative to the burden of bacteria seen in the airway, and there appears to be difficulty shutting it off. There really is, we have ibuprofen and azithromycin. We actually don't know how azithromycin works. Perhaps it's an anti-inflammatory. Maybe it has some antibacterial impact as well. But clearly we need new medications to resolve inflammation in CF without worsening chance of infectious complications. So this is what happens in the normal innate immune response. You're going along your day, you get an infection, an ear infection or cellulitis, and then your inflammatory response increases to the point that the infection is controlled. You clear the infection, and then you want to turn off the inflammatory response so that you can get back to your activities of living, and your lungs can do what they're meant to do, which is to take in oxygen and get rid of carbon dioxide in the case of CF. And so that's a normal physiologic response. And there are certain inflammatory lipid mediators that set off the inflammatory response here. And then there's also mediators that resolve the inflammatory response. And this decreases this inflammatory cells. And this is how we heal wounds and tissues. And then we want to go back to our normal state without causing immunosuppression. So I want to talk a little bit about the endocannabinoid system, because this is the target of Lenabasum. We have two receptors, CB2, which is mostly on cells, immune cells that are produced in the bone marrow, and CB1, and these are cells in the central nervous system. That's primarily where they're located, but they're also in other organs to a small amount, and in the on these immune cells that are circulating. Lenabasum preferentially activates CB2, and when it does that, it turns off or turns down the inflammatory response in the body, and then it sometimes can, to a small degree or a lesser degree, may activate the CB1, but not very much. This is the structure of the Lenabasum here. And this just shows you that when you get in this state, that Lenabasum will push things towards resolution and a return to the normal state. So before discussing the results of the current clinical trial, I want to talk about the first Phase IIa study in CF. And this was really to look at the safety, the tolerability of the study, also to understand more about the plasma concentrations of the drug. And we looked at other things, but this was mostly a study to make sure the drug was safe. Interesting, in that study, we found that the time to the first exacerbation was longer in the people who got Lenabasum than placebo. And what that means is that instead of, if you're in the placebo group, maybe you had your first exacerbation at day 45, but if you're in the Lenabasum group, maybe it occurred at day 75. So it took longer for the inflammation in your body to trigger an exacerbation. So it's kind of a weird concept. And I just wanted to touch base. So we often look at time to first exacerbation. And for first exacerbation, it was defined as people who got a new prescription for oral or IV antibiotics. And so what you see here is the placebo group was exacerbating sooner than the Lenabasum group, which exacerbated later. And this is just looking at it in a different way, but same thing. The placebo group exacerbated faster than the Lenabasum group. So that was encouraging. And in fact, when you look at the number or frequency exacerbation, so that was time to, now we're talking about frequency, you see that even in the low doses, the Lenabasum group tended to have fewer exacerbations. And in the higher group, if in 8 weeks, they also had fewer exacerbations. Now, these numbers aren't statistically significant, but they're trends in the right direction. And in an early Phase IIa study like that, you're not going to see, typically, I should say, you're not going to see huge significant differences. You could, certainly some of the Vertex drugs did, but anti-inflammatories don't necessarily show that early on. These are studies that take a lot of people, a lot of volunteers, and long periods of time to do. And this is a very quick, smaller study. And then that was backed up. We looked at markers in sputum, and you can see that this side of the curve means everything is decreasing in people who took Lenabasum. If they were increasing, they'd be over on this side. And you see that for these markers in sputum, there were some that were statistically different, but there were trends in the right direction in all the markers that we checked. So it looks like Lenabasum was associated with decrease in sputum markers of inflammation and fewer exacerbations. So this was enough to go on to a much larger, longer study, and that's where you start looking at the efficacy of the drug a little more intensely. And then after that, you move on to what's called phase 3 studies, which are the studies the FDA is really interested in. And so here's the study design. This is the 002 study. It was a 6-month study, and it was double blind, which means neither the person receiving the therapy or the study team knew which drug they were receiving. That's all coded, and only research pharmacists that aren't on site know. And there was, of course, a placebo group. There was a Lenabasum 20 milligram twice a day, so that's the big dose. And then the dose we didn't study in the first trial, kind of an intermediate dose, Lenabasum 5 milligrams. This study also went down to people with CF who were 12 years of age, where the first study was all in adults. And so when you look at, and so our primary outcome was going to be pulmonary exacerbations. So when you do a study where you're using pulmonary exacerbations as a primary outcome, you want to make sure you have enough exacerbations in the study to see a difference. And so you can enrich that for exacerbations, because the past, what happened in the last year, often predicts the future. So in order to be eligible for the study, people had to have 1 to 3 pulmonary exacerbations in the previous year, which required intravenous antibiotics, but they couldn't have an exacerbation within 28 days of the start of the study for them to get in, because they weren't probably back to their normal state. If they only had 1 exacerbation treated with IV antibiotics, they must have had at least 1 other treated with oral antibiotics. So you can have up to 3 exacerbations all treated with IV, or 1 with IV and 1 oral. So now you've got a population that tends to have exacerbations. And so how did we define what an exacerbation was? Well, let's see, let's go on to the next one. What I want to point out is that there are 447 subjects at 105 sites in 21 countries. And so how did we define what an exacerbation is? Because even clinically, we don't have hard and fast definitions. It's when a doctor or a provider decides this patient's not at their baseline, so we need to treat them with antibiotics. And so that actually was the definition we wanted to use for the study. We wanted to make this what we call a real world study. And so we thought that the best definition was when a healthcare provider decides to treat with antibiotics, whether oral or IV. We then had a meeting with the FDA. They said, that's fine, but we want you to have certain criteria to define an exacerbation. And they strongly recommended that we use the Fuchs criteria. And these are 12 things. And in order to have an exacerbation, you have to have 4 of those 12 things. So they might be increased cough, shortness of breath with exertion, a change in sputum production or quality of the sputum, sinus problems, chest x-ray findings, weight loss, fatigue. The problem with using the Fuchs criteria is it's not been validated in studies to be 100% honest. It's been used since 1993 from the original Pulmozyme studies, but it was looked at, the information was collected, and after physicians or healthcare providers were diagnosing someone with an exacerbation, it was not meant to say, hey, this is an exacerbation, you need to treat. Now it has kind of morphed into that, and there's problems with that, as I think you'll see here. For example, you can, FEV1 is one of the criteria. You can have an exacerbation based on fatigue, slight increase in cough, and so, is it a hard and fast definition? And we don't know. I don't think anybody does. And in fact, one of the things are changes on a chest x-ray, but oftentimes in the outpatient setting, we don't get a chest x-ray. So often people, one of the big things in the Fuchs criteria, people, don't have everything evaluated. Most of the things are history, some are physical findings things. So hopefully those things are picked up, but a chest x-ray, we often don't do that as an outpatient, we just treat. So, but the FDA strongly recommended that we use the Fuchs criteria to find an exacerbation. So we actually set it up as a, what we call hierarchical the analysis. One is, okay, we're going to use the FDA definition. You have to meet the Fuchs criteria and a doctor has to prescribe either oral or IV antibiotics. But we also said our secondary definition right after that is going to be just the physician decision to treat. That was it, no Fuchs criteria. The other thing is the FDA wanted us to look at the number of exacerbations as our primary outcome. So if you look up here, this rate basically means a number of exacerbations in the 6 months a person had on average in the Lenabasum group versus the placebo. We actually wanted to go in with time to exacerbation because you can enroll fewer subjects to meet that. And in an early study, we know you can't use that to get the drug approved by the FDA, but at this point, we're still trying to figure out if the drug works. And 447 patients is a lot in CF versus doing 200, right? Because there's all sorts of trials going on for people with CF. And so, but the FDA again said, nope, we want you to look at the rate or the number each person has on average and not the time to exacerbation. And we want you to use this definition. So this is what we had to go with, even though we had concerns. So this is a summary table of the study. We enrolled 181 people in the placebo group, 91 in the 5 milligram twice a day group, and 175 in the high dose group. A small number of people entered the study but decided not to. They maybe dropped out, so they did not get the drug. So you can see the percentages are fairly even across. And these are, you can forget this in the middle. These are the reasons people maybe dropped out. Some people, and you can see a tiny number, felt that the drug wasn't working. A physician, one person in each group was removed. Maybe a physician thought they weren't tolerating the drug. A handful of subjects just decided they didn't want to be in a clinical trial, and it's, their right to withdraw. Some people were withdrawn because they weren't coming to study visits or taking the drug. And you can see that's only 3 people, which is pretty impressive in such a large study. A couple of pregnancies. Some people dropped out because of side effects of the drugs, which in studies we call adverse events. We don't know if it's due to the drug or if it's just coincidence, but we capture that. And again, you see pretty small numbers, although the higher dose group was actually a little higher than the medium group and the placebo group. This is just the baseline characteristics, and you can see that they're evenly matched. The average age was in the mid-20s. There was 40 children in the placebo group, 13 in the middle group, and 32 in the high dose group. Pretty evenly split between men and women. Mostly Caucasian disease. CF is a disease that impacts Caucasians to a greater extent than other races. You can see that 1/3 of the subjects were in the US and 2/3 were outside of the US, Canada and Europe, and that'll be important later. Here, the genotype, it's the different genes fairly equally matched. The starting FEV1 fairly evenly matched. And then how about exacerbations? People had 2 exacerbations or 3 exacerbations. in the previous year, pretty evenly matched. None of these are differences that are significant. And then also the use of other drugs. So the group was pretty well randomized. So here is the very first definition. So this is the number or the rate of exacerbations per subject in the 6 months period. So if they had one, that would mean on average, they had one in that 6 month period. And this is where it becomes a little striking and we began to question. And so we're, to be honest, a little disappointed to see the results. Here in the placebo group were less, they had on average less than one exacerbation a year. But also in the two Lenabasum groups, and there really doesn't appear to be any difference, right? 0.85 and 0.9 are pretty similar. 0.76 is a hair lower. And so this was odd. This is a group of people that were selected because they had exacerbations, yet in the study, they weren't having exacerbations. And this definition was the use of either oral or IV. And when you look at IV only, the numbers are very small because half of them were treated with oral antibiotics and the patients got better. And when you look at the secondary definition, which is a physician decides to treat, you can see that it's much closer to one. We would have anticipated to be a little higher, but it is much better than using that Fuchs criteria. So this is the first clue that we probably should have stuck to our guns and insisted on that, not using the Fuchs criteria. And again, a hair lower in the middle group, but not much difference. And then when you look at IV antibiotics only, you see it's pretty similar results. And there was really, and we didn't expect a change in FEV1 in the groups. The purpose we get the FEV1 is really to make sure the drug doesn't cause the FEV1 to drop. To study a change in the rate of decline of FEV1, you're talking about, you know, a much larger, much longer study. But again, no difference. So it doesn't look like it's having an impact on lung function. It doesn't seem to be cause a problem, but you don't see much here. And how about adverse events? When you look at any adverse event, the percentages are pretty similar, maybe a hair larger in the bigger group. When you look at SAEs, and there's a certain criteria for what a serious adverse event is, one of them being hospitalization, you can see, again, fairly equal amongst the group. So a pulmonary exacerbation that causes a hospitalization is called a serious adverse event. That may or may not be due to the drug. It may just be part of CF, and we know that people who have CF have exacerbations that require hospitalizations. As far as severe, really not a lot of difference between the groups. And there were, as we noted earlier, some AEs caused some people to drop out. Again, touch higher in the higher group, and no one died during the study. You always track that whether it's the natural progression of the disease or to make sure that the drug isn't associated with that. Well, what were the AEs? Well, here's the groups. What you see here, and this is seen in other studies of Lenabasum, it's being studied in rheumatologic diseases, is headache seems to be higher in patients who take Lenabasum. You can see in the placebo group, 9.4%. In the Lenabasum, it's 12.4, 13.3. Maybe a little fatigue here in the higher dose group, a little dizziness. You know, again, slight higher in the Lenabasum group, but really not high percentages. Dry mouth, that was something we saw in the first study. That does appear to be increased on the Lenabasum group. Some night sweats. Again, these are overall low numbers, low percentages. There is a slight increase of adverse events that happens in most drug studies, but nothing here that would be concerning where you would say, oh my gosh, we should stop this trial based upon any adverse events. This is pretty typical for the studies that we do in CF. So we were pretty disappointed because the drug did not make its primary outcome. And we've been searching for an anti-inflammatory drug for many years in CF, and these results, were sobering, to be honest. But we were struggling to wrap our brains around it. It didn't make sense why people who stopped exacerbating, even in the placebo group, once they got in the study. We were, upset, I guess, because, the original outcome of timed exacerbation and physician decision to treat, I think probably would have served us better in the study. Although when you look at the whole data that I've just shown you, it's not that impressive. So, the people at Corbus, to their credit, they are very bonded to the CF community and the CF Foundation. They start drilling down into the data, and we collected data and everything. And there's a gold mine of information because we're trying to understand these results. So, they did this on their own to help inform the CF community for future drug studies. So I'd like to show you that, and we'll see some interesting things. So they started looking, they said, well, why? They said, well, how is it in the US and compared to other countries? And so they said, well, in the US, when you look at exacerbations, when you use the primary definition, it's 1.12. If you say how many of those have exacerbations, it's up to 0.72. Remember, this was right around 1. This was around, I think, 0.4 or something. And the secondary one, look at, if we use the physician decision tree, it's now up to 1.28, which that looks similar to one. That's actually a big difference. And then when you look at the IV antibiotics, it's almost double the exacerbation. So the US, that made more sense. That's what we would have anticipated. How about other countries? Well, you go, wait, well, there's one here. That's a little, that's about where it was. Here it's back down to that 0.4. This is a little down, and then this is back down to 0.51. So then what the team at Corbus did, they said, well, is there a trend in some countries? And they found a trend. And so there were 5 countries from Eastern Europe, Bulgaria, Hungary, Poland, Romania, and Serbia, that were enrolled subjects in this study. And look at this. This is really quite impressive. So these are patients. In order to be in it, you had to have at least 2, if not 4 exacerbations, right? And they only had 0.16, 0.14, 0.22, 0.16. Well, why aren't these patients, especially the placebo group, why aren't these patients, or why with this, I'll show you the placebo group, why aren't these patients having exacerbations? And so this group is part of this other countries group here. So they're probably dragging these numbers down, and they actually drag the numbers in the whole study down. All right, there we go. So when you look at the placebo group, and this is again, 130 patients, patients who had a baseline FEV1 less than 70%. Now remember, when you start making these fine cuts, the numbers get small, so you have to be careful there. But it's about 1.12, and this is everybody. And then in the 70 to 90, it's about 0.98. Now you only have 8 subjects here in the 90 FEV1 greater than 90%. So that's a small number, but they don't exacerbate much. And we decided to take all comers in the study with an FEV1 greater than or equal to 40%. We probably, looking at the data, should have had a cutoff of less than 90%. And as you would expect, the placebo group, again, overall, when you look, people who had 3 exacerbations in the first year, 1.65 rate in the study. If they had 2, they had 1.00 in the study. And if they had 1 exacerbation, it was 0.8. So overall, the placebo group is behaving as you would expect it to. They're having exacerbations, although not at the rate you'd expect, but if they had more exacerbations before the study, they were more likely to have exacerbations during the study. If you look at CFTR modulators, and you look at those people without modulators and with modulators, again, small numbers here, you got to be careful. not a lot of difference. And then if you look at US only, not a lot of difference. Now, you also have to remember, and this is a key point, that the modulators were essentially Orkambi and Symdeko. When this study started, Trikafta, lexicaftor, tezacaftor, and ivacaftor wasn't approved. Now, there were some people towards the end of the study who had started lexicaftor, tezacaftor, and ivacaftor, but that was a small number. So remember, that may have would have changed the rate, but for this study, the Orkambi and Symdeko wouldn't have had it. We know clinically it's not as dramatic as the triple combination. Really no difference if you had azithromycin or no azithromycin. People that had cultures that were positive for Pseudomonas or staph had exacerbations. If they didn't have Pseudomonas or staph, which makes sense, they had fewer exacerbations. And really prophylactic antibiotics, not much of a difference. People who take inhaled prophylactic antibiotics a little less than those who didn't. So then what happened is then you start making cuts in these groups, and you say, let's look at 40 to 90%. We're not going to look at those 8 people above 90. And again, the numbers are still small, but this is all everybody in the study. You know, 0.91, when you use the primary definition, and this is oral NIV, you see again a hair lower in the Lenabasum 5 milligram group, and there's no difference between Lenabasum and placebo. If you look at those only with IV antibiotics, again, we're down to 0.49, 0.43, and 0.45. Again, a hair lower, but not impressive. Secondary definition, you know, it's starting to see a spread here. Again, this is the physician decision to treat. It's 1.8 and versus 0.85. Now, again, interestingly, the higher dose of Lenabasum is acting more like placebo than it is the middle dose. It appears that the middle dose may be having some effect here, but the higher dose isn't. And I'll tell you some speculation about that. And this is IV only. Again, you're talking about small numbers of patients now because you're splitting them out and changing the definition. If you looked at the difference in modulators, not a huge effect with modulators on this population, but again, that's probably because we're talking about dual combination drugs versus the triple combination drugs. Now what happens if you take those 5 European countries out? Now you start to see a difference. If you look at the primary definition, look at 1.10, the higher dose group was 1.02. Now you're down to 0.75. They have a 19% decrease. Now again, you're talking 55 subjects. It's not a lot. If you look at only those with IV antibiotics, you know, again, a trend, but not significant. Secondary definition, look at that. Placebo goes up to 1.31. So that's how much those Eastern European countries change things. And the middle group was 0.96. The higher dose group, you know, again, not really any separation with placebo. If you only look at IV antibiotics, smaller, but you still see this trend here. Again, whether you're on modulators or not on modulators, here it looks like the modulators are, when you take those Eastern European countries out, because none of those patients are on any modulators, they actually look like they're improving. It's working with Lenabasum, which is what we actually predicted. And so you're seeing bigger changes in this middle group. But again, the higher dose group, which we thought we'd see a nice effect, not really much different in placebo. And how about time to first exacerbation? Now here, the last slides, when we look at rates or numbers, lower numbers are better. You want fewer exacerbations. Here, when you're looking at time, bigger numbers are better because it takes longer to exacerbate. I hope I'm not confusing people. And again, you start seeing the placebo group, when you look at the primary definition, and this is all comers again, you see the middle group is exacerbating, it takes longer to get to that first exacerbation, and the secondary definition looks even better. Modulators, those look pretty good, right? 138 versus 104, 135 versus 104, 138 versus 95 when we use that secondary definition, which we really wanted to meet our primary outcome. And so, and then when you take the Eastern European countries out, that difference gets even better. So I got 2 more slides left. This slide is kind of a summary. And then the next slide is kind of my speculation, and only my speculation. So, that's full disclosure. So this was the first Phase IIb trial to target the endocannabinoid system as an anti-inflammatory for people with CF and recurrent pulmonary exacerbations. We were disappointed that the primary endpoint was not met. We enrolled subjects who were in a refractory group of people with CF who exacerbate despite intensive background therapy. Subjects in the placebo group, when you looked at them, had the expected clinical course once you took out the 5 Eastern European countries who unexpectedly had low rates, right? Like, how do you, to be in the study, how would you go from all these exacerbations to hardly? And there was one site that they enrolled 20 subjects, and in those whole 20 subjects over the 6 months, they had 1 exacerbation. So something's going on there that doesn't make sense. And I don't have enough knowledge of those countries personally. Subjects in the US had the highest exacerbation rate. Does that mean that they're sicker in the US? No, we actually think that it means that physicians at CF care centers in the US are more aggressive and more predisposed to treat changes in symptoms and signs with antibiotics because, over the last several years with the quality improvement initiatives out of the foundation and the focus, and we understand the downsides of exacerbation and its impact on lung function and overall quality of life, we've become much more aggressive to pull the trigger on treating symptoms that are mild, maybe with oral antibiotics, or jumping right in with IV, even small changes in FEV1. And this was a shift. We didn't always do that, but I think that's what it means. When we did a post-hoc analysis, means it's analysis that you do that wasn't planned. It occurs after the study, and there was, it looks like something's going on in that 5 milligram twice a day dose group. That might not be going on in the 20 milligram group, and I'll tell you some speculations there. Overall, the drug was safe and well tolerated, and the safety profile, as it was in the first study, is unchanged. So here are my speculation, really. So there's high doses. The question is Lenabasum also activating that CB1 agonist, and you're getting some pro-inflammatory effect? And people who do medical toxicology and pharmacology also point out that this is a receptor agonist, and it typically is a U-shaped curve, in that there is an ideal dose. And if you go higher, and I don't quite understand this, it's called hormesis, Hormesis, that you don't see the same effects. And so maybe we're up on that curve with that higher dose. We are going to do further analysis. So, I didn't get to show you any curves because this data, we're still trying to make sense of it. It just came out in early October. We want to do Kaplan-Meier curves like I showed you from the first study, and we want to do statistics with Cox proportional hazard. The other thing we want to look at is, does Lenabasum help resolve and return to baseline in FEV1? I'm sure many of you are aware that some patients get better faster than others, and some patients don't get all the way back up to their pre-exacerbation FEV1. We just didn't have time to look at that. We did these patients had visits every month, so we can actually do that. And it'll be interesting to see. We also collected biospecimens. We haven't had a chance to run the assays to look at those biomarkers to see if there's any change in biomarkers. And of course, hindsight's 2020. If I had to do over again, or if Corbus had to do over again, we should have stuck to our guns and kept our primary outcome as time to exacerbation, and use the definition of exacerbation as physician decision to treat with antibiotics and not Fuchs. Because then we could have made a case in a phase 3 study on why our concerns about Fuchs, but there seems to be, just because of tradition, this faith in the Fuchs, and I don't know if it's justified. We probably, certainly in the future, we would perform trials in North America, and select Western European countries, Australia, because I think the standards of treatment there in those countries is more similar, and I think there's other parts of the world where we don't actually know how they treat exacerbations. I do believe that we're still going to need anti-inflammatories, even in the days of highly effective modulators. And remember, 10% of people won't have modulators yet. And what about people who have modulators but already have more advanced disease? They're still going to exacerbate. And we don't know long-term the impact of modulators alone on inflammation. It probably does have anti-inflammatory effect. I think they actually work in synergy with anti-inflammatory drugs, but that actually makes it harder to study, because how do you tease out? And then the question is, and this is the most concerning, what outcomes are we going to use in our future studies in this new area of highly effective modulators? We know overall modulators stabilize and preserve the FEV1 for longer periods of time, and that's a great thing. They reduce exacerbations, and so if they reduce exacerbations, people who are taking highly effective modulator won't have as many exacerbations. So what do you use to see a change in your drug? I don't know. I mean, it's a good problem to have, but I still think we're going to need anti-inflammatory drugs. And again, the earlier and earlier these drugs get started, you can imagine if a child gets started at 6 months of age, they may not exacerbate for many, many years, but do they need anti-inflammatory drugs? They may. We just don't know. So then the question is, will this be one of the last big anti-inflammatory trials in CF, or there's several hundred people in the study? That, to me, as someone who's interested in this and studies this, is a bit sobering. What are we going to use for outcomes? Are we going to use biomarkers? In the first study, you saw sputum. Well, in the highly effective modulators, after a couple of weeks, people have, tell us they can't produce sputum anymore for their first time in their lives in many, many years. So how do you study a biomarker unless you go down and do a bronchoscopy? And people be willing to do a bronchoscopy, and our blood marker is a good reflection of what's going on in the lung. And those are all questions that we don't know the answer to, and that are imperative for us to figure out. I think we will have to go with a biomarker, but we're going to have to figure out how that drug works on a molecular level, and then analyze specific products that are directly associated with that drug's mechanism of action, rather than maybe the downstream impacts of inflammation, like what goes on in the sputum. So again, we're disappointed the trial didn't work out the way we hoped. We've learned a lot, and there is a mountain of data that we can learn, and Corbus, to their credit, despite the results, as very willing to share this data with the CF community. And they're doing the analysis they can, but we're going to have to go down and really dissect this data out and figure it out, because it's going to impact how we do clinical trials for anti-inflammatories in the future, but also other therapies. And so, I wish it would have turned out the other way. I think everybody does, but this is where we are now. So with that, I'll stop there, and I'll be happy to take questions. I don't know, Siri, if you've been monitoring the chat or.
Yes. we do have questions. Thank you so much. That was fascinating. And I have a whole page of notes, but we may have to talk offline because it's too long. But we do have questions in the box saying two are somewhat related. How long a study might Lenabasum need before it sees statistical significance? Should it have been in a longer drug trial like 1 year instead of 8 months?
Well, so in retrospect, yeah, we didn't know how, so when you're designing a trial, you say, how many people do we need to figure this out? And so you go back and you look at what's been published. So we worked with the consultants in the biostatistics core at the Therapeutics Development Network and said, listen, if we use exacerbation as our outcome, how long do we have to study patients or volunteers, and how many volunteers do we need? And so you look at that data to make a determination. And again, the rate of exacerbations is lower than you would have predicted based upon that data. And we're not sure why that is. Now, we do know people when they're in trials and, they tend to take better care of themselves because they know they're being monitored. So that could play a role. And certainly we weren't anticipating the issue with the European countries. But, if you do a study for a long year, like the original ibuprofen study went 4 years. Well, it's hard in a disease that we call an orphan disease like CF, where you don't have millions of patients. You have 30,000 people in the US, 70,000 worldwide, to tie patients up in those studies. Because once you're in a study, you have to drop out of the study if the drug becomes available that you want to take, because it's a new therapy and it changes the result. So fortunately, most of the people, when Trikafta came on board, most people were through it, and the ones in the study were coming out of it. A few got enrolled afterwards, but they were on the Trikafta for the study. And so, patients aren't going to agree to be in a study that long. So, yeah, obviously, I think, if we had more patients in a longer study, you probably, wouldn't, who knows? It maybe wouldn't have shown a difference. You don't know. But then you get to the point, was it even feasible to do a study of 2,000 people for, a year and a half? And I think most people would say it's hard to find someone to commit to that.
Another question was, what's going on with those 5 Eastern European countries? And I guess that also triggered for me, like, what is the life expectancy for people with CF in those countries with, I think you explained about diagnoses that were more aggressive in the US, but what?
Yeah, so the truth is, when we saw this data, that's the problem. When I said, how are those countries chosen? And they're like, well, we went to a consultant who said, these are the countries. And the problem is we just don't know what they're doing there, and we don't know what they have available, right? Like in some countries, they may not have inhaled antibiotics or inhaled Dornase alpha, and we don't know anything about their survival in those countries. I could speculate. I probably shouldn't, though. So it's a great question. It's a question we asked ourselves.
Okay, the Fuchs criteria.
Mm-hmm.
How is something like that developed, and how can a group of physicians and researchers such as yourself with all this experience come up with a criteria that becomes the standard?
Yeah, so again, that was developed not as say, we're going to use this and move forward. It was like, okay, we have these exacerbations. Let's figure out why people in the study or the other clinicians decided to treat with the antibiotics. And so that was just developed. Genentech, who developed Dornase alpha, was just trying to figure out, because to be honest, everybody's doing a little bit different across the US. And still that happens sometimes today. The foundation has worked so hard to standardize care. But back then, and then we just didn't know. So everybody's trying different things. And so in the study, they wanted to look at it in the after fact, the post hoc analysis to figure it out. I can tell you that many of us, people smarter than me, have been trying to convince the FDA that that is not the way to go. That was not developed as a prospective definition. It's been used a lot, but this is a study that we're using exacerbations, not saying, oh, we want to know why people were prescribing it. And so I think many of us feel, and that's why we chose it. We actually put together consultants when we were designing the study. I put together this amazing team of individuals who were really experts in exacerbation, and we agreed that physician decision to treat with a new antibiotic is the best definition, and to try to force a square peg in a round hole. There is work being done on part of the Therapeutics Development Network, even CF Foundation, and then also investigators to try to change this thought construct or this with the FDA that we have to use the Fuchs. There's other definitions, and the Fuchs is no better than any of those others. It's just been around the longest and it's more popular. We don't know if it's any better.
For people who were in the trial who did think they had drug, not placebo, and were feeling so much better, who may be nervous now about possibly there not being access to this in the near future, if trials are conducted in other disease groups that Corbus is working with the same drug, is this something where patients with their physicians would possibly have access through that route if it didn't pan out? Yeah.
So that's a discussion to have with your physician. Once a drug gets on the market, a physician can write a prescription for an off-label use. Now, whether the insurance company will be willing to pay for that off-label use, that's another story. But in pediatrics, until recently, when I got into pediatrics, most drugs that we used were never studied in children and were never approved down to children. Like ceftriaxone was a very popular antibiotic to treat community-acquired pneumonia in the hospital back in the 80s and 90s, and even still used today. It has never been studied in children. It's an adult drug, and we said, well, we need something. We're going to use that, and we could write for it. Things are getting a little more strict about that. So if the drug gets approved for one of the rheumatologic conditions, first of all, that's a discussion with the provider. I would find out, first of all, what group you are in, to make sure you're in, the group, and you can get that. You can ask your site. The data is still being analyzed, so they won't break that blind yet, but eventually they'll break that blind, and your site should send you a letter, because that's what the TDN does. People want feedback. Well, what the results show and what group was I in? And so you should find that out. And then that's a discussion, and I'm not advocating either way on that you should have with your provider.
Okay. I'm so grateful to you, because I know people have a lot of questions. I have two more, and then we'll let you go back. How does Lenabasum's Phase 2 trial results compare to the Celtaxis, I'm going to slaughter this, Acetaminophen Phase 2 trial?
Yeah, the Celtaxis, the big difference, we did not participate. I personally did not participate in that study. In Cleveland, I thought the Celtaxis team was first rate, very good. They chose as their outcome, instead of exacerbations, FEV1 decline, because that's what ibuprofen showed. The problem is when ibuprofen was studied in the late 80s and early 90s, there were no drugs for CF. And so people were losing 3.5 percentage points a year. So they might go from 96 to 92 to 88 in a year. That's huge. We're down to like 1 and less than 1, and it's going to be even smaller with the triple combination drug. And so they chose FEV1 as their primary outcome for that study, and they didn't hit it. And so there's a lot of weight put on the primary outcome. That study had also 440 patients. Interestingly, though, they found that if they looked at people who had an exacerbation, so this is one of those post hoc analyses, in the year prior to entering the study, at least just one exacerbation, and looked at the exacerbations in the study, those who got their drug had a reduced number of exacerbations than those who didn't. So that's another drug. I'm going to be honest, I worry not just the Lenabasum or the Spinostat drug, but other anti-inflammatories, that because of study design issues, if we haven't thrown the baby out with the bathwater on some of these. And the problem is once you get tagged as not meeting your trial results, it's hard to go back and get those drugs because it is expensive to to develop drugs. So many of them fail and they're so expensive, these trials. And for someone to say, well, this is a drug that failed, let's go back and take a look at it, invest even more in money into it and see if we change maybe the drug itself and a little bit, and we change the study design and we do this. There's these approved drug will take $500 million up to $2 billion eventually with all this drug development to get it out there in 13 years, and it's not protected, for long periods of time. So it's often hard to get someone to say, I think this is a drug that we should go back and take a look at and see if we can and do it. And so that's one of the problems with when the drugs fail, and I just sit there and go, man, have we missed some good opportunities? I don't know.
And is there hope for us who have been watching this with such optimism that once, I know it's still so early and looking at all the data, but is there hope to be able to go back and sift through and continue on?
Well, so we're going to go back and we're going to really go into this data and do a deep dive and look at the biomarkers and the studies, see what we can see and what we can learn from it. I have to say we presented these results to the leadership of the CF Foundation, and they were very gracious. They supported this study financially, but also in other ways. Of course, they wanted it to be more impressive. But in my mind, they are not stopping looking for anti-inflammatory drugs. They did admit it that it's going to be hard to do the traditional clinical trials we've always done. And I said, so are you telling me what, I'm a little concerned about where my career is headed. They go, no, this is the exciting part of your career. You're going to be busy for a year. So there is a commitment from the foundation. They're not giving up because they're worried about that 10%. And they're worried about, until there's a one-time cure, the long-term impact of inflammation, even if it is as a much smaller level than it with the modulators than it has been. So there's still a commitment. It's just going to be harder, I think.
Well, we are right up on the hour, and I cannot thank you enough for sharing your time, your expertise, your perspectives on this. We have been so curious about this trial, and this explains so much for many of us. If people do have questions that we were not able to get to, may I email them to you and see if we can get answers to them? And I thank you. I thank our sponsors, Vertex Pharmaceuticals, Gilead Sciences, Genentech, Chiesi USA, Viatris, and the additional support for today's presentation from Corbus Pharmaceuticals. So thank you all for joining us, and I wish you a good rest of the day. Thank you.
Thank you.